Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Have you ever wondered if there might be a way, maybe a gentle way, to nudge our brains back towards better health, especially when facing something as daunting as Alzheimer's disease? Imagine, if a treatment that wasn't invasive could actually help restore the connections that are so fundamental to our memories or thinking. Well, today we're diving deep into a really fascinating new study that explores exactly that possibility. It looks at how low-intensity brain stimulation might do something quite remarkable for our neurons. Our main source today is a recent health news piece from Medical Express, published just June 30th, 2025, and it digs into a really groundbreaking scientific paper in neurophotonics. The research itself comes from teams at the University of Queensland and the University of Tasmania. So our mission today is to unpack what they found, what the core findings are, understand what it all means, the implications, and explore what it might signal for the future of managing Alzheimer's. We'll try to give you a clear picture without, getting lost in overly complex jargon.
Right. And Alzheimer's, it's such a critical issue. We're talking about a neurodegenerative condition,
Yeah.
and it really does profoundly affect millions of older people across the globe. At its heart, Alzheimer's messes with the brain's communication network. Your brain cells, the neurons, they're constantly talking to each other at these specific
The
points.
synapses.
Exactly, the synapses. These are the crucial connection points. And the brain normally has this amazing ability to adapt and change these connections, grow new ones, trim old ones, make them stronger or weaker. We call that synaptic plasticity.
So it's like the brain is constantly rewiring itself, optimizing things.
That precisely. That dynamic process is fundamental for how we learn, how we form memories. But in Alzheimer's disease, this vital synaptic hugely. And right now, there's no effective cure. We have treatments, but they mainly manage symptoms. That's why exploring new approaches like this is just so important.
Okay, so let's talk about the technique at the center of this study. It involves something called repetitive transcranial magnetic stimulation. It's often shortened to rTMS by researchers, so maybe we can stick with rTMS for now.
Sounds good.
So rTMS, it's a non-invasive method, right? Uses electromagnetic pulses to target specific brain areas from outside the head.
That's it.
And it's not entirely new, is it? We know it can boost synaptic plasticity in healthy brains, and it's already used for some other conditions, neurological or psychiatric ones.
That's correct, yes.
Yeah.
rTMS definitely shows therapeutic promise. The challenge, though, especially with Alzheimer's, has been figuring out why some people respond better than others. The responses can be quite variable. And the precise mechanisms, like what's actually happening at the microscopic level with the neurons, well, that hasn't been fully pinned down. So that variability and that gap in understanding how it works deep down, that's really what this new study was digging into. They wanted to see those tiny changes.
Got it. So this specific research we're looking at, the researchers, they investigated how rTMS affected these crucial connection points, the synapses, in the brain cortex of mice that actually had Alzheimer's-like dementia. Their goal, and this is quoting the corresponding author, Dr. Barbora Filipova, was to essentially count and measure the changes in what are called synaptic axonal boutons,
Right.
comparing the Alzheimer's model mice to healthy mice, because, as she put it, synaptic dysfunction is a key mechanism in the disease. So understanding these changes is critical.
And understanding axon boutons is key here. Think of the axon as the long output cable of a neuron. These boutons are tiny, specialized structures, like little knobs, either at the very end of the axon or sometimes dotted along its length. These are the spots where the synapses actually form, where one neuron passes the signal to the next. So you can imagine any changes to these boutons, to their number, how stable they are, how they function, that's going to have a big impact on brain connectivity, how well the whole network communicates.
So they're like the critical communication hubs. And did they look at different types of these boutons?
They did, yes. They focused on two specific types of excitatory boutons, the ones involved in activating other neurons. They looked at terminaux bouton and en passant bouton.
Terminaux and en passant. Okay, what's the difference?
Think of terminaux boutons as being right at the end of an axon branch. They make sort of very direct, often local connections, like a dedicated endpoint.
Okay, like a specific address.
Sort of, yeah.
Yeah.
Whereas en passant bouton, en passant meaning in passing, are like little beads strung along the length of the axon. An axon might pass by several other neurons, and these boutons allow it to form connections along the way, not just at the end.
So more like stops along a route.
Exactly, connecting potentially more distant regions or multiple targets. And to actually see these tiny structures, which is incredibly difficult, they use a really advanced imaging technique called two-photon imaging.
Photon. Okay.
This allows you to look deep into living tissue, in this case, the brain of a live mouse, and get these incredibly detailed 3D images of individual axons and synapses. You can literally watch things changing over time.
Wow, that's amazing seeing it live. And the mice they used weren't just standard lab mice, were they?
No, that's crucial. They used a specific genetic model. It's called the APPPS1 XY1GFPM strain. But the important thing is these mice are engineered to develop features very similar to human Alzheimer's disease, like amyloid plaques.
Right, the plaques we hear about.
Yes. And another key thing, some of the neurons in these mice were also engineered to produce a green fluorescent protein. So their axons literally glowed green under the microscope.
Ah, making them easy to track.
Precisely.
Hmm.
It allowed the researchers to track the changes in those specific axonal boutons with incredible accuracy over time in the living brain. So the team watched these boutons, how many there were, how many appeared, how many disappeared. They monitored this at 48-hour intervals over 8 days, both before and then after a single session of rTMS.
And then compared everything back to healthy, normal mice as a baseline.
Exactly. The wild-type mice, that comparison is key.
Okay, so they've got the model, the high-tech imaging, the timeline. Let's get to the findings. What did they see before they applied any rTMS? What was the baseline situation?
Well, interestingly, at the start, before any stimulation, they found that the overall density, just the sheer number of both terminaux boutons and en passant boutons, was actually pretty similar between the Alzheimer's model mice and the healthy mice.
Oh, so it wasn't that they simply had fewer connections to begin with.
Not in terms of raw numbers, no. But, and this is really the critical point, the turnover rate was significantly lower in the Alzheimer's model mice.
Turnover, meaning how quickly they change.
Exactly. Turnover reflects the dynamics, how often new boutons are formed, existing ones are lost, or they get restructured. It's a measure of plasticity, really. And this dynamic activity was much lower for both types of boutons in the mice with Alzheimer's-like changes. And this reduction in dynamism is likely linked to that amyloid plaque buildup, which is characteristic of Alzheimer's. It suggests that even though the basic wiring might be there, its ability to adapt and change, which is essential for learning and memory, is already impaired.
So it's like the network is becoming rigid, less flexible.
That's a good way to put it, less adaptable.
Okay, so then came the rTMS, just one single low-intensity session. What happened then? This seems like the really exciting part.
It really is. This is where things got dramatic. After that single rTMS session, the turnover rate of the terminaux boutons shot up significantly. And this happened in both the healthy mice and the Alzheimer's model mice.
Both groups saw an increase.
Yes, but the effect was much more pronounced in the Alzheimer's group. They saw the biggest changes about 2 days after the stimulation. For the healthy mice, turnover increased by about 88%.
Pretty good boost.
Yeah, but for the Alzheimer's model mice, it was a massive 213% increase.
Wow, 213%. That's huge.
It's an extraordinary increase in the dynamic activity of these connections. Now, it's important to note this big surge was temporary.
Ah, okay. How long did it last?
It peaked around day 2 and then gradually returned back towards the pre-stimulation levels by the 8th day of observation.
So maybe needing repeated sessions for a lasting effect.
That's exactly what it suggests for potential clinical use, yes. But the really striking thing is what that temporary boost achieved. In the Alzheimer's model mice, that massive increase actually brought their terminal bouton turnover rate up to levels that were comparable to the healthy mice before they got any stimulation.
So it effectively normalized their turnover rate, at least temporarily.
It suggests a potential restoration? Yes.
Okay.
Like it kick-started that synaptic plasticity again, pushing those connections back towards a more healthy, adaptable state, even if just for a while.
That's incredibly promising. Did both types of boutons respond this way?
No, and that's another really interesting piece of the puzzle. Only the terminaux boutons showed this significant increase in turnover after rTMS. The en passant boutons didn't really change.
Huh. So it's specific.
It seems to be, yes. It suggests the mechanisms might be, as the researchers put it, cell-type specific. Maybe rTMS preferentially affects these more direct terminal connections with the circuits they're part of. And this increased turnover wasn't just about gaining new boutons or losing old ones. It was driven by increases in both. Just a general ramping up of the dynamic remodeling of these specific synaptic sites.
Okay, so let's connect the dots. What does all this mean for us, for people potentially facing Alzheimer's? What are the real-world implications here?
Well, this is genuinely significant. It's the first time, really, that we have direct evidence, seen in vivo in a living system, that these presynaptic structures, the boutons, respond to rTMS in both healthy brains and in a model of dementia. And as Dr. Filipova pointed out, we already know that synaptic problems are deeply linked to cognitive decline and dementia. We also know rTMS is used elsewhere. So putting that together, these findings really highlight, in her words, rTMS's potential as a powerful addition to currently used Alzheimer's disease management strategies.
So maybe not a cure in itself, but a potentially powerful tool to add to the toolkit.
Exactly. It opens up a new avenue for treatments that are targeted at the synapse level, treatments that could potentially improve quality of life for people with Alzheimer's.
How might that look for a patient?
Well, imagine if you could use this non-invasive stimulation to help boost the brain's own ability to maintain its connections, to keep that plasticity going. For patients, maybe that translates to slowing down the cognitive decline, holding on to memories longer.
Improving daily function.
Yes, enhancing overall quality of life by directly tackling some of that synaptic dysregulation that drives the disease. It's about potentially keeping the brain's network healthier, more adaptable for longer.
That sounds incredibly hopeful. But as with any exciting new research, we have to ask about the limitations, right? And what needs to happen next.
Absolutely. It's crucial to be realistic. This is foundational work. So first, we need to know if these effects are specific to the brain region they studied, or if rTMS affects these boutons more widely across the brain. That needs checking.
Okay, scope matters.
Definitely. Second, future studies really need to try and measure the actual electrical activity of the neurons in real time while the animals are awake and maybe even doing tasks, like learning something new.
To see the functional effect.
Exactly. To understand the physiological response during active brain function. And the big one, as we touched on, is the single session versus multiple sessions.
All right.
This study used just one shot of rTMS. Clinically, rTMS is almost always given in repeated sessions over days or weeks. So future research absolutely has to look at the effects of repeated stimulation. That's critical for translating this to potential human therapies.
That makes sense. So to kind of wrap up where we are, this deep dive shows that even a single low-intensity rTMS session seems to hold, well, pretty remarkable promise. It looks like it can boost neuron health, maybe even restore some synaptic plasticity in an Alzheimer's mouse model, specifically by jump-starting the activity of those terminaux boutons, those direct connection points.
That's the core finding, yes.
And while clearly more research is needed, especially on repeating the stimulation, it really does seem to open up some fascinating new possibilities.
It really does. You know, just imagine a future where we could use personalized brain stimulation tailored to an individual to help preserve that incredibly complex dance of our neural connections. Not just slowing symptoms down, but maybe, potentially, helping to restore some of the very fabric of memory and thought. This study offers a really tantalizing glimpse of the brain's amazing capacity for plasticity, for change. And it'll leave you wondering, doesn't it? What other incredible discoveries about the brain's ability to adapt, maybe even heal itself, are just around the corner?
That's a powerful thought to end on, a lot to think about there. Thank you for joining us on this deep dive into this exciting piece of brain health research.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.